XYZ cultivar or Zikla, effective in ABRXO1. I'm going to abbreviate that as XOC, so it'll be a lot easier to explain. So XOC causes bacterial leaf streak in rice, and this is a very devastating disease because it affects rice in Asia and Africa. And we know that rice feeds up to 1/3 of the world's population. So XOC, this bacteria, has a lot of effector proteins. One of them is ABRXO1, and that's what my project is mainly about. The reason why we only look at ABRXO1 as an effector protein is because the mechanism and function is very little known about it. So this is what makes this project very interesting. But what is known about ABRXO1 is that one, it's secreted by many plant pathogens, not only XOC, but also two, that it helps trigger resistance in resistant plants and virulence in subtainable plants. So that means that in plants that are trigger resistant plants, all the cells around it die if XOC is secreted in it. And the susceptible plants, ABRXO1 helps XOCs thrive and last longer in the plants. So it kind of is helping it out, which is interesting because we don't know how that happens. Also, XOC uses a type 3 secretion method to secrete ABRXO1. And what a type 3 secretion method means is that you have the bacteria right here, and it's pretty much injected a needle directly into the host membrane. So unlike any other bacteria, it goes directly into it. So the purpose of my project is to identify the sections of ABRXO1 that help with suppression of yeast in the fragment. So I have two goals. One of them was to get the fragments that are toxic for yeast and also fragments that are expressed in yeast. So to do that, I designed primers that were cloned, and then we added the gateway P enter vector into these cloned ABRXO1 primers. After those fragments, we subcloned them and added the BD into them. And then here's the breakdown of my results. That the whole fragment of ABRXO1, 420 amino acids, doesn't allow anything to grow, but these three subfragments do. So I focused on this subfragment as my yeast 2 hybrid. And in these subfragments, we added AD, which has cDNA, and then we combined those two things of the AD and the cDNA of the rice, and that's how we get the yeast 2 hybrid. And so we went through a selection method to find the interactors, and through the selection method we found that there's 17 interactors, and of those 17, 12 had predictive function, and 7 of those ones had unique function. So up here, these were the 7 interactors that we had. We had many different ones, but the most important ones were the ones that affected glycolysis, because glycolysis is needed for all living organisms, prokaryote and eukaryote, and glycolysis helps with metabolism. So it's affecting the food source for many of these rice plants. Also, we found that it affects chloroplast and mitochondria areas, which means it's targeting the energy for these rice. So in conclusion, these 8 interactors are targeting chloroplast and mitochondria, and we have hypothesized that ABRXO1 may target glycolysis, which is a food mechanism. So the whole purpose of this project is hopefully to figure out and target specific areas and make resistant plants of rice, so this disease of XOC could be kind of decreased and have a resistance plant.